This is a cluster of 4 or 5 cells around the outside surrounding 2 cells in the middle that are dark because they're not expressing the fluorescent protein. The green cells that we can see here are the migratory cells, and they extend and retract protrusions, and we can even see them changing position so that the leader cell is not always the same. So the second one's also a control. Now the border cells are expressing the photoactivatable form of RAC. It's activated by 458 nanometer light, and we're not illuminating with that. And so this is just a control to show that the construct in the absence of the activating wavelength of light is inert. You can see the cells migrate very similarly. They more or less look the same as the ones we were looking at in the first movie. Number 3 is the first real experiment here. This is a really fun movie because the normal direction of migration would be from the upper left to the lower right corner, and these cells start moving in that direction. But then Chabot shines the light off to the side, and now the whole cluster by 45 minutes is off to the side of the egg chamber. And this is something that you would just never observe in either of the control situations. They migrate actually all the way to the side of the egg chamber. And when they get there, he tries to continue to get them to go down the side of the egg chambers. But in fact, all they do is just keep changing positions. And that told us that there's some constraint within this tissue that's not permitting the cells to migrate in that particular location. And we don't know the nature of that constraint because we never even knew it existed until we did this experiment. When he changes the direction of illumination now, the entire group again now moves in the direction that he's illuminating. So this simple experiment really told us that focal activation of RAC is sufficient to set the direction of migration, not just for a single cell, but for this entire cluster of cells. And this is in a natural environment where they're surrounded by lots of signaling pathways. Figure 4 is another control, and what they did was make one point mutation in the light sensing domain that renders the protein insensitive to light. That allows us to control for everything else we're doing to this egg chamber. If we just want to control for all of those things that we're changing when we do this experiment, then we can use this light insensitive version of the protein. And so it's just one amino acid different from the one in movie number 3, and yet we see that when we illuminate off to the side, the cells do not move to the side. They continue their normal migration path from the upper left to the lower right. Here we wanted to see if we could the cluster move in the completely opposite direction compared to its normal direction of movement. So at the beginning of this movie, he's actually illuminating at the natural front of the cluster and causing them to migrate in their normal direction. And in fact, what happens is that they migrate faster than they normally would in that direction. But we can see the very dramatic extension of protrusion directly under the laser light. And then he stops illuminating the front and now starts illuminating the back. The cells realize something's different. Just about instantly, the cells stop protruding at the front. And we can see fairly rapidly some protrusion at the back, but you don't get backward movement until after a delay. We think that what's happening is that the cluster is initially polarized, and the front cell knows that it's different from the back. Now we start illuminating at the back, and it takes some time for each cell to adjust to its new identity in order for the whole cluster to move backwards. But eventually it does. And so we are interested in learning the nature of the communication between the cells that allows each cell to sort of understand at any given point in time if it's a front cell or a back cell, and then what the nature is of that repolarization event, which seems to take some time. This is another clever thing. The idea here is that they fused the light sensing domain to an inactivated form of RAC. If we illuminate at the front of the cluster, we see that the cluster extends many fewer protrusions. But what eventually happens is that the cells at the side and back of the cluster start extending protrusions. So all we have to do is inactivate RAC in one part of the leading cell, and the whole cluster gets confused. That was actually surprising, because if each cell were sensing the gradient of chemoattractant, then you might imagine if one cell couldn't sense it, that the majority of cells would know which way to go. They'd still go ahead and go. But at relatively short timescales, this confuses the whole cluster. It really says that these cells are sensing direction collectively, and they basically choose which direction to go in based on which cell has the highest level of RAC activity.